Welcome to Dr. CM Edcast, your go-to resource for medical learning. Through discussions, case studies, tutorials, and debates, we help you learn, grow, serve.
You know, when you think about it, the heart is pretty incredible. Most of the muscles in your body need a direct command from the brain to do anything. But the heart, it's different. It has its own internal, self-contained electrical system. It's this beautiful synchronized network that generates and coordinates every single beat, all on its own. So let's take a journey through that intricate symphony. So here's our game plan. We're going to follow the complete journey of a single heartbeat, from the very first spark to the final pump. We'll start with the heart's electrical wiring, then see where that spark comes from. We'll trace its path, figure out why there's a crucial built-in delay, and then finally, we'll watch it all come together in that powerful contraction that keeps us all going. So, first things first, how is the heart even wired for this? Well, the whole system is built from these really specialized muscle cells. But here's the cool part. Their main job isn't to contract. They've actually evolved for one primary purpose: to create and conduct electricity. Okay, take a look at this. This is basically the electrical map of the heart, and these are the key players in our story. The whole journey kicks off right here, at the SA node. That's the heart's natural pacemaker. From there, the signal travels to this critical checkpoint, the AV node. Then, it's sent down a kind of high-speed highway called the bundle of His, which splits off and uses these Purkinje fibers to deliver that final message to the main pumping chambers. All right, so where does that initial spark, the one that starts every single heartbeat, actually come from? Well, it all begins in that tiny little cluster of cells we just saw, the SA node, which is tucked away in the wall of the right atrium. The real secret to the heart's rhythm is something called the pacemaker potential. See, most muscle cells just sit around waiting for a command, but these SA node cells are totally different. You can think of them as constantly, slowly filling up with an electrical charge. They just automatically build up this charge, it's called depolarization, until bang, they hit a tipping point. And when they do, they fire. They create the spark. This incredible ability to start on their own is called autorhythmicity. And you can see the difference so clearly right here. On the left, a typical muscle cell's charge stays flat, just waiting for a signal. But look at the pacemaker cell on the right. See that constant, gradual upward slope? That's because the cell is naturally a bit leaky, letting sodium ions slowly trickle in and build that charge. As soon as it hits that threshold line, it fires, setting the pace for the entire heart, somewhere around 60 to 100 beats per minute. Okay, so the SA node has fired. The spark is lit. Now what? Well, that electrical impulse doesn't just stay put. It immediately begins a rapid journey across the heart's upper chambers, the atria. As this electrical wave spreads like a ripple through the muscle fibers of both atria, it triggers them to contract. And this contraction is what gives the blood that final squeeze out of the atria and down into the lower chambers, the ventricles. It's basically priming the pumps for the main event. And this first part of the journey is unbelievably fast. We're talking just 0.03 of a second. In the blink of an eye, that signal has already crossed the atria and arrived at its next major stop, the AV node. Now, when the impulse gets to the AV node, it doesn't just get a green light to pass right through. Nope. This little structure acts as a crucial gatekeeper. It intentionally imposes a very deliberate and absolutely essential pause in the signal's journey. And that pause lasts for about 1/10 of a second. Now, I know that doesn't sound like much, right? But in the life of a heartbeat, that fraction of a second is everything. So why the holdup? Well, it all comes down to timing. This delay gives the atria enough time to finish their squeeze, to make sure the ventricles are completely full of blood right before they get the signal to contract. Without this pause, the pump would be really inefficient. It would be like trying to fire a cannon that's only half loaded. You just wouldn't get that powerful, effective result. All right, the delay is over. Now it's time for the grand finale. The signal is finally unleashed to the heart's main pumping chambers, the ventricles. And this last phase, well, this is all about speed and perfectly synchronized power. The impulse just rockets down the bundle of His, which splits into what are called the bundle branches. These run along the interventricular septum. That's just the muscular wall that separates the right and left ventricles. So the very first part of the ventricle to get activated is this central wall. From those bundle branches, the signal is handed off to the Purkinje fibers, and this is this huge tree-like network that spreads all throughout the ventricular walls. These fibers conduct that impulse incredibly fast to make sure that all of the ventricular muscle cells contract in a near perfectly synchronized wave. And here is just a brilliant piece of biological design. The signal travels all the way down to the bottom, or the apex, of the heart before it spreads up through the walls. This forces the ventricles to contract from the bottom up. I mean, think about squeezing a tube of toothpaste from the end. It is the absolute most efficient way possible to wring every last drop of blood upward and out into the body. And these numbers right here, they tell the whole story. Just look at the speed in the AV node. It's a crawl. That's our deliberate critical delay. Now compare that with the Purkinje fibers. They are an absolute superhighway, conducting that signal up to 200 times faster to guarantee that massive, all-at-once contraction of the ventricles. So let's just put it all together, from that very first spark at the SA node all the way to the completion of the ventricular squeeze. This entire elegant sequence takes just over 0.2 of a second. It's an absolute masterpiece of biological engineering, repeated with every single beat of our lives. And over an average lifetime, this perfectly timed electrical symphony will conduct itself more than 3 billion times. It's an almost unfathomable number of flawless performances. And that really leaves us with this final thought. The near perfection of this 0.22-second journey is what keeps us alive. But that perfection is also its greatest vulnerability, because a tiny disruption, a single misstep anywhere in this pathway, a signal that's blocked or even slightly delayed, that's all it takes to throw off the rhythm, to cause an arrhythmia, which is really a central focus of modern cardiology.